muscle is called smooth muscle because it does not have striations. Now, what does striations translate to? What's creating the striations in skeletal muscle? What creates the striations in skeletal muscle? I think I'm hearing it in the back. The sarcomere, right? The sarcomere. So we don't have sarcomeres in smooth muscle, and so they're called smooth. They're found in our internal organs, liver, heart, stomach, not heart is going to be cardiac muscle, but in our blood vessels, in our uterus, the bladder, things like that, and our blood vessels, okay? They're going to be under involuntary control, which we know, right? Smooth muscle is one of those tissues innervated by the autonomic nervous system. And they're also going to be different in their shape. So because they're not fibrous or elongated like we see with the sarcomere, they're going to be more of the spindle-shaped cell. Okay, we'll look at what that looks like in a little bit. But very different from this fibrous filamentous structure that we see with cardiac muscle, which is striated, and that we see with skeletal muscle, which is also striated. The size is going to be much smaller than skeletal muscle, so much, as you can see here, these cells are much smaller than the entire muscle fiber here. And again, we don't have sarcomeres, but we do have actin and myosin. We do have that interaction between actin and myosin to contract these types of cells. We also have the presence of dense bodies, and dense bodies are synonymous with the Z line. Okay, they're the point of anchoring the actin filaments together, and when these smooth muscle cells contract, we're going to have those dense bodies coming closer together, very similar to how we saw those Z lines coming closer together. Okay. Okay, so let's look at this here quickly. So we have smooth muscle. First off, we're going to mention that the contractions here are much longer, much slower. And I'll talk about why in a little bit. Okay. Also, these cells are much smaller, right? About 1/10 the size of skeletal muscle cells or skeletal muscle fibers, okay? We also see no striations, meaning that there are no sarcomeres. And we know that there is still actin myosin, which means that there's still the sliding filament model happening here. Okay, so these are the properties that we know are true for smooth muscle that kind of distinguish them from skeletal muscle. Let's look at what a smooth muscle cell looks like. First of all, it's spindle shape, as you mentioned. It's not going to be elongated or fibrous the way the skeletal muscle cells are. Okay. We also know that it's going to be innervated by these muscle fibers that have varicosities. Okay. Don't forget to make that connection. We do not have a nerve terminal here. We do not have an axon terminal. What we have are these varicosities. This is a smooth muscle example. Now, along the muscle cell, we still have the interaction of actin and myosin. Okay, so I'll just draw these here. So at the point where actin and myosin meet, or sorry, at the point where different actin molecules meet, this is called the dense body, okay? And it's kind of getting messy here, but you get the point. So where these two actin cross, this is called the dense body. And as I mentioned earlier, this is synonymous to the Z lines or the Z discs, okay? Now we also have the myosin or the thick filament. So along each of these actin molecules, we have myosin, sort of just hanging out in the middle here, okay? And I'll talk about how this contracts and how this brings about muscle contraction. So obliquely arranged, that's sort of the difference here, that this is arranged crisscross, sort of obliquely with the actin molecules. So we have our thin filament, which are these black lines. That's actin. Oops. And then we have the thick filament here. Now, as you can imagine, because of this oblique arrangement, when the myosin head is energized, which I'll talk about, the entire cell is going to sort of crumple down on itself because of this oblique arrangement, okay? So myosin is going to slide across the entire length of actin here, and the entire cell is going to sort of crumple down together because of that oblique arrangement, all right? So just to reiterate here, some of the differences, we have varicosities, not the axon terminal, not the neuromuscular junction. We have the dense bodies, which is sort of similar to the Z lines. When this contracts, these two dense bodies get closer together, okay? We have myosin, which is sort of moving obliquely along actin. And then we have these dark lines here, which are representing thin filaments or actin, okay? So we still see the sliding filament model. We still see actin and myosin interacting, but in a very different way, okay? It's even regulated in a different way. We'll talk about that. But we do have them sliding past each other, so there is still that sliding filament model. And when they do so, this crumples down this cell, almost like crumpling up a piece of paper, and makes it really small. And that is how a smooth muscle cell contracts, okay? All right, any questions on that bit? Any questions? Okay. So hopefully that will make more sense looking at this image, okay? So as we can see here, we have the sliding filament model. We have a longer contraction because myosin is moving across the entire length of actin, right? We have that thin filament, we have the myosin sort of sitting on top of the thin filament, and they were connected at these dense bodies. And so when the contraction happens, myosin moves across the entire length of actin, and that's what gives us a longer, slower, more sustained contraction, okay? We also have this oblique arrangement, such that the cell kind of crumples down on itself because of that spindle shape.